Kia ora team, welcome back to the 2.5 Genetic Variation and Change 4-credit external video series. This is video 11. In this video, you'll be learning about linked genes. And by the end of this lesson, you should be able to describe linked genes and the possible gametes produced when genes are linked, explain why linked genes are not affected by independent assortment, explain how linked genes are affected by crossing over, and discuss how linked genes lead to unexpected phenotypic ratios. So at this point, you've learned that the law of independent assortment states that the alleles of a gene segregate independently of other alleles from other genes, but only if those genes are on non-homologous chromosomes, so if they're not on the same chromosomes. I think these two diagrams clearly show this. Because gene A and gene B are on different chromosomes, their alleles are able to be shuffled by independent assortment, resulting in 4 possible gametes. There is an equal chance for each gamete to be made, and the laws of segregation and independent assortment are crucial to understanding this lesson. You've also learned how to do a dihybrid cross. And you also now know that if 2 individuals that are heterozygous for both genes are crossed, and the 2 genes in question are unlinked, the vast majority of their offspring should have the dominant phenotype for both traits, and only a very small proportion of their offspring should have the recessive phenotype for both traits. But this 9:3:1 ratio only works if the 2 genes are unlinked, because they can assort independently. So what happens if genes are linked? Linked genes are genes that are located close on the same chromosome. Because they're physically joined, they tend to be inherited together. Since linked genes are on the same chromosome, it is impossible for linked genes to independently assort. As a result, the gametes produced show less genetic variation than unlinked genes. This should help you visualize that. In this scenario, gene A and gene B are located very close together on the same chromosome, meaning that they're linked. Since gene A and gene B are on the same chromosome, it is impossible for the big A and the little a alleles to assort independently of the big B and the little b alleles. Because the big A allele is physically joined to the big B allele on the blue chromosome, those 2 alleles will segregate into the same gamete. The same goes for the pink chromosome. Because the little a allele is physically joined to the little b allele on the pink chromosome, those 2 alleles will segregate into the same gamete. As a result, there are only 2 possible gametes, and there is an equal 50-50% chance for each of the gametes to be made. So when we put the linked genes and the unlinked gene scenario side by side, we can see that linked genes, so genes on the same chromosome, lead to less genetic variation in gametes because the big A and the little a alleles cannot assort independently from the big B and the little b alleles respectively. The big A allele is stuck with the big B allele, and the little a allele is stuck with a little b allele. The unlinked gene scenario leads to more genetic variation because the alleles aren't physically joined and can independently assort. So there's an equal chance in creating gametes that have the little a and the big B alleles, and gametes that have the big A and the little b alleles. But in reality, we can get this. We can get linked genes with crossing over. Remember that during crossing over, the 2 inward-facing non-sister chromatids of homologous chromosomes interlock at the chiasma, so here's the chiasma, and they exchange corresponding or matching segments, shuffling or recombining the maternal and the paternal alleles, creating more genetic variation. So remember, crossing over is a chance event. It happens at random. So in the rare chance crossing over happens between the paternal blue chromosome and the maternal pink chromosome, we can create recombinant gametes. These increase genetic variation because these gametes are different to both of the parents. So this parental gamete is big A, big B, identical to the blue chromosome, and this parental gamete, little a and little b, is identical to the pink chromosome. Whereas this recombinant gamete, big A, little b, and this recombinant gamete, little a, big B, are both different from the combinations the parents had, therefore creating genetic variation. I just want to put these two scenarios side by side so you can see the differences between the gametes made between linked genes and unlinked gene scenarios. You can clearly see that when genes are linked, almost all of the gametes will have a parental combination of alleles. So these are the parent combinations of alleles, and 96% of all the gametes have the parental combination. And only a very small percentage of the gametes will have undergone crossing over and form the recombinant combination of alleles, so only about 4%. This is because the likelihood that a chiasma will form exactly in between gene A and gene B is low. The closer the genes are on the same chromosome, the less likely a chiasma will form in between them, and the less likely recombinants will form. On the flip side, you can clearly see that when genes are unlinked, all four gametes have the same likelihood of occurring because of independent assortment. So when linked genes are close together, crossing over is less likely to occur. The distance between the two genes is so short that the chance of a chiasma forming exactly between gene A and gene B is low. The closer the genes are together, the less likely crossing over will happen and the less likely recombinants will form. But when linked genes are far apart, crossing over is more likely to occur. In this case, genes A and B are still linked because they're still on the same chromosome, but the distance between the two genes is so long that the chance of a chiasma forming between gene A and gene B is now much higher. The further apart the linked genes are, the more likely crossing over will happen and the more likely recombinants will form. Let's look at the two linked gene scenarios side by side. On the left-hand panel, we can see that when linked genes are close together on the chromosome, crossing over is less likely to happen, so recombinant gametes are very rare. The exact number, 2%, as what's on the slide, doesn't matter. What matters is that the percentage of recombinant gametes is much lower than the percentage of parental gametes. So a combined 4% versus a combined 96%. Now the percentage of recombinant gametes will actually increase as the distance between the linked genes increases and the likelihood of crossing over increases. On the right-hand panel, we can see that when linked genes are far apart on the same chromosome, crossing over is more likely to happen, so recombinant gametes will be more common. The percentage of parental and recombinant gametes in this scenario is 25%, meaning that in this scenario, gene A and gene B are basically at the tips of the chromosome as far apart from each other as possible. So how do linked genes affect phenotypic ratios? If genes are unlinked, you would expect a phenotypic ratio of 9:3:3:1. But why do we expect this 9:3:3:1 ratio? In a dihybrid cross, if the two genes are on different chromosomes, then alleles sort independently. Each parent produces 4 types of gametes in equal frequency, so 25% for each type of gamete. And the proof is in the 4x4 Punnett square that you complete. You'll see that you get the 9:3:3:1 ratio. But linked genes break this phenotypic ratio. If genes A and B are on the same chromosome, the original pairs of alleles seen in the parents will be more common than recombinants. The recombinants will only appear if crossing over occurs between the two genes during meiosis. The parental gametes will occur at a much higher frequency, and the recombinant gametes will occur at a much lower frequency. So if genes are linked, you would expect a phenotypic ratio of something like 9:1:5, where the recombinant phenotypes are lower than the parental phenotypes. In linked gene scenarios, the recombinant phenotypes are rarer than the parental phenotypes. It all still has to add up to 16, because there are 16 squares in the Punnett square. So those missing numbers get added to the parental phenotypes in the ratio. So if genes are linked, the parental phenotypes are overrepresented in the phenotypic ratio, and the recombinant phenotypes are underrepresented in the phenotypic ratio. Well done, you've survived this linked genes lesson. So by now you should be able to describe linked genes and the possible gametes they produce, explain why linked genes are not affected by independent assortment and how linked genes are affected by crossing over, and you should also be able to discuss how linked genes lead to unexpected phenotype ratios. Thanks for watching, and I'll see you in the next video.